design your sequence, you get to actual sequencing, right? However, what is the limitation of like sequencing and IFRP? Here, what is the limitation of that? Anyone have any idea? Anyone want to try to give an answer? Because, for example, if you have one patient walk in, and then okay, I think I'm suspected that this patient will have, for example, cancer of platelet, that's what I say, and then I will send the blood for the test, and then send it test to the lab, and I actually searched in the literature saying, okay, it's got mutation that being identified before. So I went to NCBI and then searched that gene and then design primer. Okay, what if it's got like some syndrome that I try to my best to describe this and try to match it in the literature. That is, okay, no one has actually published that mutation being recorded associated with this condition before. Can I do this? No. Right? Because you got to know the sequence to be able to design primer. Right? To be able to do, because this is based on that you have something in your mind that, okay, some other people saying in the genome of the trillion gene, in the trillion base pair in the genome, this particular region is actually being associated to particular phenotype or particular disease. So therefore, we want to look at that particular region to see whether we find that particular mutation in our patient or not. So if you don't have pre-existing information, so you can't do this, right? So you can actually use the what they does in the past would be like the mapping by the gene mapping by using the repeat area in the genome, because in the genome 3 billion base pair for the gene area is actually just only 2% of the genome is actually in the gene area. Other than that, it's got like repeat area. And in the past, people used what I call the short tandem repeat to signify this is a mark on the map of the genome. And then we don't know what gene causing that yet, right? We may have, oh, this guy coming and then have this disease. Oh, a few years later, sibling come with the same syndrome. Oh, it must be something to do with genetic, right? So they got like the whole genome, measuring that. Let's say this is a whole genome. This particular region, okay, have 10 repeat of the STS, 20 repeat, 30 repeat, and then you try to find that which repeat would actually go with the phenotype. For example, if that doesn't really have a phenotype, and then they use a model try to predict that what type of the inheritance that getting on this disease. For example, whether it's actually autosomal recessive, dominant, X-linked, or whatever, right? And then, okay, we think this region in the genome would actually associate to that, and then they do the sequencing for the whole family, try to do the linkage analysis. And then they may find later that, okay, this particular gene will actually link to the disease. Okay, but that is in the past, right? I think the technology, the new kind of sequencing that will actually allow us to have the throughput for the sequencing is called next generation sequencing, where you can actually sequence the whole genome in a very fast manner, maybe just 10 gigabytes, I think, that you have like maybe 5 or 10 gigabytes, you have one genome being done within days, and then with analysis maybe a month also. But in the past, for the whole genome project, sequencing it takes quite a long time to complete the first map of the whole genome project. So I think the one of the key milestones that I mentioned, that we're talking about the short tandem repeat with the mapping, and then people talking about the SNP, single nucleotide polymorphism. Like every 1,000 base, it will be some position to be marked, rather than you do it like some random area of the STS. You have the SNP area that being reported to be associated. However, you still need like some sequencing that to be able to get through the whole sequencing. That when they start having the new platform of the next generation sequencing. Did I do something wrong? And that will be like in 2005, and actually the first one actually launched is actually quite expensive, but now it's as it's become more popular, as it's become cheaper and cheaper and cheaper, and now it's up to the level that can be affordable by the small lab. And that depends on the levels that you want to look at, how strong the association is. You can do whole genome sequencing, which means that you can sequence for the whole genome. You have a lot of data, genome. We'll talk about how sequence later on, but just talking about the big picture, just the whole genome sequencing. Or you can do just only the whole exome sequencing. So the exome sequencing is you pick up just only the exon area. Gene is only 2%, right? And then you get exon and intron in the gene. And then if you capture just only the whole exome sequencing, which will be the region that mostly will encode for the coding region for protein, you actually have to pay less to get information that you may have more chance to get information in this area. So it depends. So for the platform itself, how do we do this? For the next generation sequencing, you have DNA, right? You're going to make a library. You have to share it with the nuclease, and you add in the adapter or the barcode just to tell that this one from one sample, and then after you have that barcoding, you amplify it up and do the sequencing. Along the way, you actually testing the quality of the DNA or RNA for the library preparation as well, before you may measure the quality by looking at some of the surrogate markers, make sure the quality is there. After you're making library, you also make sure you get homogeneous population of the size that you want to share, and then adding index into it as well. And then you run on the sequencing itself. This is just an example of the platform that being used is from the Illumina, just quite a popular platform to use. We use the bridge, what called bridge amplification. So you have a region of adapter that will actually complement to the static phase of the chip that have a lot of nucleotide, and that after having your sequence binding hybridized to the other part of the sequence, and you have the amplification going one direction. So you have amplified product of this one end, and then after you having that amplification going, you actually have the sequence do the reading from the different nucleotide as it go. Okay, as your nucleotide would actually tag it with the different fluorophore. Every single amplification you capture the sequence, and once you get information of the sequence of interest of the gray one, you can actually get them to align with the reference sequence of the genome. As you can see here, the short sequence of every single step you capture, and then you get information of one to the other, A, T, C, G of each spot on the pre-filter on the filtering part of that. And then once you get the sequence of all those being aligned, you have different short sequence you align onto to the reference sequence. And then you looking at the read by using the bioinformatic analysis, try to align those, and then try to see whether you have walk base at that reference sequence. So at the end, you will looking at the depth number. It means that how many A sequence that you can read from the one particular position of the reference sequence. You may get the depth of 100x. It means that at particular position, it being have a short sequence, 100 sequence being aligned on this, and that will give you a sort of the reliability of whether that sequence is actually a real G. However, by having this, this is sequencing for the whole genome in a very short time, but you still have to confirm it with the Sanger sequencing, because it's got some error when you actually have this new technology as well to do it as well. You need to design a primer again when you actually identify SNP or base change that you think it matters to your condition. After you design a primer into a particular region, and then you confirm it with the Sanger sequencing, and then you may want to confirm it in another patient sample with the same condition as well, to see whether it's actually causing disease or associated with disease. So this new platform, well, relatively new, it's not new anymore. It's like 10 and a bit years now. It's actually allow you to sequence and do association in a quicker manner. You can send samples outsourced to the company, and you have the information with the analysis in a month or so time, and can associate with your particular condition of interest. Okay. So that is that one. I think I'd like to touch on the last part of this, which I think is not included here. Is the germline and somatic mutation, right? Because we talk about the mutation that we can detect, right? That using PCR. That PCR will actually give you a different size product, and you can actually design the IFRP. You can do the sequencing just to identify the base change. That could be nonsense mutation, or it could be missense mutation. But when you deal with the germline mutation, it's actually quite clear when you're looking at the band pattern, like This one is clear that is homozygous mutation allele here, or heterozygous, you were talking about 0%, 50% and 100%, right? But in a field of cancer in particular, we got something called somatic mutation. Somatic mutations mean that the mutation that is actually having a base change later in your life. That could be, you have some mutation that is prone to have base change before that. For example, if you have cancer in the eye, let's say retinoblastoma, you have cancer, but maybe not a good one. Let's say, okay, platelet cancer, talking something that I may be more familiar with. Platelet cancer, it doesn't mean that in the cell that, in all cell that you have in your body, you have mutation. But you can actually have a cell at some particular source that would actually have a mutation. If you look at the skin, take the skin, and then try to find a mutation in the DNA, you may not find it. But if it's platelet mutation, okay, take the blood out, and then I know it's some population of the cell, of the stem cell, that would actually have a mutation. But it's not all cell of the stem cell, because it's actually just a clonal evolution of some cell. Let's say if you have 100 cells being taken from the blood, maybe 10% of the cell you have mutation. So if you have your assay to detect this for that particular disease, you may have to look for something that have what we call tumor burden. So you're not saying dealing with something that is 0%, 50%, and 100%. You're talking about 10% burden, or 50% burden, or 20% burden, which means that you may need to put a perspective of the sensitivity of your assay into that as well, when you're actually looking at the detection. Okay, I think I will stop here. And so I will change that to be the other one. Let's stop here. That you can scan and give comment, and if you can go and then give the look at a questionnaire and then can fill up that would be much appreciated. And I think we will see you again on the protein lecture, I think after RNA and then protein. Okay, thank you very much for today. Sorry again for the messing up with your schedule. Thank you.